This week is week 11. We're going to be talking about biodiversity. Just as a heads up, it's going to be a little bit longer than our typical lectures. Got a lot to go through, not only with biodiversity, but also what's going to be due next week and to round out the semester. So let's get into it. Biodiversity can refer to a few different things. You've got species diversity. We see this every day where we compare birds to beetles, to humans, to all different types of species. Genetic diversity refers to the variations within those different species. And then you have ecosystem diversity, with just comparing tropical rainforests to deserts, to coral reefs, so on and so forth. The general trend of diversity here is that there's a lot of diversity around the equator. So in the Amazon jungle, the coral reef over on the Philippines, those are biodiversity hotspots. And biodiversity tends to taper out as we move towards the poles. So Antarctica and the Arctic are less biodiverse than some of the habitats and ecosystems around the equator. This is just sort of a snapshot to indicate exactly how much diversity there is on this planet. You see that there's bacteria, plants, fungi, fish, amphibians, reptiles, birds, mammals, and then humans are all the way at the very bottom here on the right-hand side. And we have not been on this planet for very long. The first things to arise were bacteria, and then plants developed, and then fungi, and so on and so forth. So here's some diversity by numbers, according to science. We have discovered around 4,000 species of bacteria, animals in two different categories, vertebrates and invertebrates, around 52,000 for vertebrates, 1.2 million for invertebrates, and then plants around 270,000 different species of plants. And total described species is around 1.7 million. However, scientists estimate there's more likely closer to around 14 million different species on this planet, a majority of which have been undescribed by science. So let's start off with plant diversity. There's two major types of plants. You've got your monocots and your dicots, and they differ in a few different ways. One being the embryo, the leaf venation or the veins on the leaves, the stems, the roots, and the flowers. In monocots, you see that there's one cotyledon, and a cotyledon is sort of like an embryonic leaf of the plant. It's the kind of the first leaf to show up after a seed germinates. And in monocots, mono meaning one, you have one embryonic leaf seed. And then in dicots, you have two. The veins in leaves of monocots are usually parallel, where the veins in dicots are usually net-like. Within the stems of monocots, you see these vascular bundles, usually arranged in complex structures and not really organized, whereas in dicots, you see the vascular bundles usually arranged in a ring. The root system also differs between monocots and dicots. In monocots, you have more of a really fibrous root system, where in dicots, there's usually one large taproot and other roots extend out from that main root. And then the flowers in monocots usually show up in multiples of 3, whereas in dicots, the flowers usually show up in multiples of 4 or 5. Continuing on, there's diversity with different pollen types and how that pollen is spread. Pollen can be considered the male gametes of the flowers. Plants can self-fertilize, but they ideally would prefer to cross-fertilize with different members of the same species just to increase that overall genetic diversity. And pollen can be dispersed a few different ways by sticking to animals like we see here with bees and butterflies. Pollen can also be dispersed by wind. If it's really light, the kind of strong breeze comes through, can blow the pollen in all sorts of different directions, hopefully landing on a female flower of the same species. The female part of the plant can be seen through fruits and seeds. This is often considered the ovule, which contains an ovum, which is the female gamete. Ideally, the plant wants its offspring, both pollen and seed, the female gametes, to go far, far away. That way it decreases the competition of its offspring close by. And like I mentioned, there's different ways that plants can be dispersed. You have wind dispersal, you have animal dispersal. This is not only by the pollen or the female reproductive system sticking to animals, but also animals digesting seeds and depositing them elsewhere. Then you have water dispersal. A good example of that is by the coconut. You know, palm trees usually are seen on tropical islands. Coconut is nice and heavy and can float on the water, drift away to other islands where then that coconut will start sprouting up. So let's think about the avocado and how it might be dispersed. So as we all know, avocados have a huge seed, also known as the pit, that they grow right in the center of their fruit. If we think about this, this is kind of a really heavy pit, not really good for wind dispersal, maybe for water dispersal, since avocados did originate in Central America. There's a lot of rivers down there. But if we look at the fossil record, we notice that there used to be huge giant animals, one of which being the giant sloth that ate avocados. So seeds of the avocado pass through the digestive tract of these large animals, and then they were deposited in other areas as the animals moved throughout the ecosystem. So we can consider the avocado as a tree or a species of plant being dispersed by animals through their digestive tract. Now let's look at animal diversity. So this is just a schematic of all the different phylums of animals. You've got annelids, which are your earthworms, arthropods, which are your insects, your bees, your wasps, your butterflies, moths, browzoans, which are a marine, kind of like sponge-like group of animals, chordates, which makes up all the vertebrates, so your frogs, your reptiles, your chimpanzees, humans, Cnidaria, which are corals and jellyfish, echinoderms, which are sea stars and starfish, sea urchins, sand dollars, mollusks, which are your snails and your squid, octopus are all mollusk, nematodes, which are parasitic worms, platyhelminthes, which are marine worms, rotifers, which are planktonic creatures, and then sponges, which are dominantly live in the ocean, and they are some of the earliest known animals on this planet. But a majority of animals on this planet are considered beetles. Beetles make up roughly 25% of all species on this planet. Beetles are insects, so they're within the phylum arthropods, but there's a huge diversity of beetles on this planet. 1 in every 4 species that you come across would be considered a beetle if you took the plant or the animal diversity as a whole. From this, we can see that a majority of animals on this planet are in fact invertebrates, so animals that do not have a backbone, and sponges down here represent the earliest known or the most basal lineage of invertebrates and animals as a whole. On the other end of that, you've got a small portion of animals that are considered vertebrates within the phylum Chordates or Chordata, and these are animals with a backbone. And fish are the earliest ancestors of modern day vertebrates. We are part of this group. Horses, chimpanzees, elephants, turtles, frogs, anything with a backbone is considered a chordate. So let's talk about how species are arranged. How are they grouped together? There's a few different ways that we group species, and this could be through homologous and analogous structures. Homology is considered to be shared characteristics due to a common ancestor, does not always have to have the same function. So let's look at an example of the dragonfly and the atlas moth. We see that both of them have 6 legs, both of them have 2 pairs of wings, both of them have compound eyes, And this is because they're both insects, so they shared a common ancestor relatively close to each other down the evolutionary tree. On the opposite of that is analogous structures. And these are shared characteristics due to common function, not common ancestor. So if we take a look at the dragonfly and the heron, we see that both of them have wings and both of them are used for flight, but they do not have a common ancestor, right? Heron is a bird and the dragonfly is an insect. So different different ancestry, same function is analogous structures. Similar ancestry, but not always the same function is homologous structures. Homologous structures are not always super obvious, but the vertebrate skeleton helps us to determine how animals are grouped. So if we look at different vertebrates, like we do with birds, humans, dogs, wolves, the limb structure of these creatures are very similar. The forelimbs, the leg bones, the rib cage, the pelvis, the skull are all arranged in a similar fashion. And this is because we all share a common ancestor at one point down the line. Not all of the forelimbs or the leg bones are used in the same way. So for dogs, it might be used for super fast running. The forelimbs in birds might be used for flight. In fish, they're used for swimming. And frogs are used for jumping. So they're used differently. They all have the same layout because we do share a common ancestor with all these creatures. We're all considered vertebrates. And then an example of analogous structures could be quills. So quills are considered hollow spines used for protection from predators. That's their function. And we can see this is an example of convergent evolution because we see that mammals like hedgehogs and porcupines have quills. Plants like cacti have quills, and invertebrates like sea urchins also have quills. Now, these don't share a common ancestor at all. Sea urchins are echinoderms, hedgehogs and porcupines are chordates, and plants or cacti are plants. But they have the same common function, which is protection from predators. And they're made of different materials. So this is an example of analogous structures that leads to convergent evolution, which is same function of different structures, but different ancestry. So why do we care about biodiversity? Why is it important? Well, genetic diversity is extremely important for agriculture. The more diversity you have within a certain crop, the better you'll have a crop yield and crop development. Healthy ecosystems need to be diverse to maintain their natural resources. And of course, humans like to travel to these beautiful places like Yellowstone and Yosemite and coral reefs in the Philippines and so on and so forth. So tourism is a huge factor when looking at biodiversity. And then the more diverse an ecosystem is, the more likely it's going to have human application through engineering or new medicinal compounds. But as a whole, there's different ecosystem functions and different ecosystem services that are tightly tied to the biological diversity of that ecosystem. So in this graph here, we can see that we have biological diversity on the x-axis and ecosystem function on the y-axis. And we see this nice relationship to where if we increase biological diversity going from left to right or right to left, we see that the ecosystem function also increases. But what is an ecosystem function? Well, it's the primary production of the ecosystem, it's the nutrient cycling, it's the decomposition. These are all functions that ecosystems have. And these functions lead to different ecosystem services. So the production of food, water, wood, regulating climate, regulating disease are all ecosystem services that humans benefit from. And we see that the higher diversity an ecosystem has, the better the ecosystem functions and the better overall society thrives. So it may not be super important that one species of frog goes extinct here or there, but if we continue on this trend, we're definitely going to hurt ourselves. And so biodiversity is incredibly important to make sure that ecosystems continue their function and services for humans to benefit from. So, but biodiversity loss is a natural process. Extinction is a natural process. If you remember back from a few lectures where we talked about survival of the fittest, This occurs naturally over many, 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 many years. Extinction is a gradual process. But humans, we've had a very short time on this planet. We have increased the rate of extinction by nearly 1,000 times. And this is not good. You know, biodiversity is extremely important, like I talked about. Extinction is a natural process, but the fact that we're increasing the rate of extinction by over 1,000 times is it's not a good sign for us down the line. Just by this increased extinction, we have put roughly 30% of all amphibians vulnerable to extinction. Roughly 20% of all mammals, roughly 12% of all birds are now vulnerable to extinction because of our actions. So that is all I have on biodiversity. Let's talk about some of the assignments that are due next week and what's upcoming for the rest of the semester. First off, you have an exam next week. Again, it's going to be available on Canvas from 12 to 1:40, and it covers lectures from weeks 9. 10 was an instructor help week, so there's no lecture there. And then this week's lecture 11. Additionally, you'll have the algae final report. I initially told you guys it's going to be due on your class day, whether you're Wednesday or Thursday. However, because we have an exam, I'm going to extend that deadline to Friday, April 9th at 5 p.m. So you have a few extra days there to get that in. I know it's a lot to study for the exam as well as write this report. Remember, the outline of what the report should look like is on Canvas under Files. I think the assignment title or the document title is called BIOL 100 Final Reports. And then we're going to start our sourdough manipulative experiment. So for this, what you're going to do is you're going to follow the exact same procedures as the first sourdough experiment, except this time you're going to pick something to manipulate, whether it be the pH of the water that you use, different flour, etc. You can check page 27 of your lab manual for different examples of what to manipulate. Remember, based on what you manipulate, it has to be testable because you're going to write a hypothesis. The hypothesis needs to be testable. So again, take data on the height of the sourdough in your jar. You want a minimum of 10 days of observations. And then instead of turning in a separate report for this, what you're going to do is you're going to, again, turn in a final report, including the title, intro, methods, results, discussion, to me on week 14. Not only for this manipulative sourdough, but also for the previous sourdough experiment that we did. Again, if you have any questions, please feel free to email me. We're wrapping up the semester quickly here. Things are moving quite fast, so I want you guys to make sure you're good to go and no questions before the time comes where we finish everything and turn everything in. So that's all I got for you guys today. Let me know if you have any questions. I'll post this on YouTube and send it around Canvas as well.